Today we're going to discuss the topic, Asthma Inflammatory Process, by Professor Swaminathan Kirahanam. Contact mail ID, profsamiacademy@gmail.com. Next slide, early, late and chronic stages of asthma inflammation. First, the early phase is initiated by IgE antibodies that are sensitized and released by plasma cells. These antibodies respond to certain triggers like infections, allergies, smoke, fumes, some medicines, emotions and pollution. The result is airway inflammation, increased vascular permeability, mucus secretion, bronchospasm, and wheezing. These events are referred to as the early asthmatic response because they occur within minutes. Second, the late phase asthma attack. About 60% of asthmatics who experience the early phase will experience this late phase. Mediators that initiate late phase reactions are thought to be derived from resident mast cells activated by IgE and allergen, or from T cells that recognize allergen-derived peptides. Such T cells may be either resident at, or recruited to, sites of allergen challenge. The late phase reaction is clinically defined as bronchoconstriction, a fall of 15% or more in forced expiratory volume, FEV1, or 50% fall in airway conductance, which occurs 12 hours after the early phase reaction to inhalation of allergen challenge. While symptoms may feel similar, there are more notable breathing difficulties due to a recurring airway blockage, obstruction, in this phase. Third, chronic asthma, CA. Asthmatic inflammation develops when the sequential interaction of inflammatory cells with resident cells generates a cascade of events that contribute to the chronic inflammation and clinical manifestations associated with the disease, including further inflammation, airway smooth muscle spasm, bronchospasm. Next slide, 10 important components for airway inflammation in asthma. 1. Role of epithelial cells in airways inflammation. 2. Role of eosinophils in airways inflammation. 3. Role of lymphocytes in airways inflammation. 4. Role of Th1 and Th2 cell imbalance in airways inflammation. 5. Role of mast cells in airways inflammation. 6. Role of alveolar macrophages in airways inflammation. 7. Role of neutrophils in airways inflammation. 8. Role of fibroblasts and myofibroblasts in airways inflammation. 9. Role of inflammatory mediators in airways inflammation. 10. Role of adhesion molecules in airways inflammation. Next slide. 1. Role of epithelial cells in airways inflammation. Bronchial epithelial cells as a barrier to participating in mucociliary clearance and removal of noxious agents. However, epithelial cells also participate in inflammation by the release of eosinoids, peptidases, matrix proteins, cytokines, and nitric oxide. Epithelial cells can be activated by IgE-dependent mechanisms, viruses, pollutants, or histamines. In asthma, epithelium may influence the sensitivity of the airways to various provocative stimuli. Epithelial cells also may be important in the regulation of airway remodeling and fibrosis. Next slide. 2. Role of eosinophils in airways inflammation. Eosinophils play an effector role in asthma by the release of pro-inflammatory mediators, cytotoxic mediators, and cytokines. Circulating eosinophils migrate to the airways by cell rolling, through interactions with selectins, and eventually adhere to the endothelium. Eosinophils enter the matrix of the membrane. Their survival is prolonged by interleukin-5 and granulocyte macrophage colony stimulating factors. On activation, eosinophils release inflammatory mediators such as leukotrienes and granule proteins to injure airway tissue. Next slide. 3. Role of lymphocytes in airways inflammation. Mucosal biopsy specimens from patients with asthma contain lymphocytes, many of which express surface markers of inflammation. There are two types of T helper CD4 cells. Type 1, T helper, Th1, cells produce interleukin-2 and interferon gamma, both essential for cellular defense mechanisms. Th2 cells produce cytokines, interleukin 4, 5, 6, 9, and 13, that mediate allergic inflammation. It is known that Th1 cytokines inhibit the production of Th2 cytokines, and vice versa. It is hypothesized that allergic asthmatic inflammation results from a Th2-mediated mechanism, an imbalance between Th1 and Th2 cells. Next slide, 4. Role of Th1 and Th2 cell imbalance in airways inflammation. Allergic reactions are mediated by antibodies of the IgE class. IgE-mediated allergic reactions are said to be atopic. T and B lymphocytes play an important role in the production of IgE. There are two types of T helper cells, Th lymphocytes, designated Th1 and Th2. People who are prone to develop allergies, that is, atopic people, are believed to have a higher ratio of Th2 and Th1 cells, and this is believed to be an important factor in their tendency to produce allergy-mediating IgE antibodies. These observations are clearly relevant to asthma since biopsies of the bronchial mucosa of patients with asthma have an excess of activated Th2 cells. Many authors take these observations to indicate that an imbalance in Th2 and Th1 plays an important role in the development of allergies and, specifically, asthma. Next slide, role of Th2 versus Th1 cytokine in allergic asthma. Asthma is caused by an increase in Th2 and a decrease in Th1 cytokines. Th1-type cytokines tend to produce the pro-inflammatory responses responsible for killing intracellular parasites and for perpetuating autoimmune responses. Interferon gamma is the main Th1 cytokine. Th2-type cytokines mediate the activation and maintenance of the humoral, or antibody-mediated, the immune response against extracellular parasites, bacteria, allergens, and toxins. Th2 cells mediate these functions by producing various cytokines such as interleukin 4, 5, 6, 9, 13, and 17E. Next slide, role of Th2 versus Th1 cytokine in allergic asthma. Activated Th2 cell-mediated asthma is caused in part by the secretion of interleukins, example, interleukins 4, interleukins 5 and interleukins 13. These cytokines stimulate B-cell activation, plasma cells and IgE secretion. Th2 cell cytokines and IgE activate cells of the innate immune system, example, eosinophils, mast cells, etc. This leads to release the histamine, hepain, serotonin, etc. Next slide, 5. Role of mast cells in airways inflammation. Mast cells are found throughout the walls of the respiratory tract. Mast cell degranulation is important in the initiation of immediate responses following exposure to allergens. Once binding of allergen to cell-bound IgE occurs, mediators such as histamine, eosinophil and neutrophil chemotactic factors, leukotrienes C4, D4, and E4 prostaglandins, platelet activating factor and others are released from mast cells. Mast cell degranulation is believed to be an integral cause of exercise-induced bronchospasm following either drying or cooling of the airways. Next slide, 6. Role of alveolar macrophages in airways inflammation. The primary function of alveolar macrophages in the normal airway is to serve as scavengers, engulfing and digesting bacteria and other foreign materials. They are found in large and small airways, ideally located for affecting the asthmatic response. A number of mediators produced and released by macrophages have been identified, including platelet activating factor, leukotriene B4, leukotriene C4, and leukotriene D4. Additionally, alveolar macrophages are able to produce neutrophil chemotactic factor and eosinophil chemotactic factor, which, in turn, further the inflammatory process. Next slide, 7. Roles of neutrophils in airways inflammation. High numbers of neutrophils have been reported to be present in the airways of patients who died from sudden-onset fatal asthma. This suggests that neutrophils may play a pivotal role in the disease process, at least in the sudden-onset fatal cases and in some patients with long-standing or corticosteroid-dependent asthma. The neutrophil also can be a source for a variety of mediators, including platelet-activating factors, prostaglandins, thromboxans, and leukotrienes, that contribute to airway inflammation. Next slide, 8. Role of fibroblasts and myofibroblasts in airways inflammation. Fibroblasts are found frequently in connective tissue. Human lung fibroblasts may behave as inflammatory cells on activation by interleukin-4 and interleukin-13. The myofibroblast may contribute to the regulation of inflammation via the release of cytokines and to tissue remodeling. In asthma, myofibroblasts are increased in numbers beneath the reticular basement membrane, and there is an association between their numbers and the thickness of the reticular basement membrane. Next slide, 9. Role of inflammatory mediators in airways inflammation. Lung mast cells are an important source of histamine. This histamine is capable of inducing smooth muscle constriction and bronchospasm and is thought to play a role in mucosal edema and mucus secretion. Arachidonic acid can be broken down by the enzyme cyclooxygenase to form the prostaglandins. A further breakdown product, prostaglandin D2, is a potent bronchoconstricting agent. Prostaglandin I2 contribute to inflammation and edema owing to its effects as a vasodilator. Thromboxane A2 is produced by alveolar macrophages, fibroblasts, epithelial cells, neutrophils, and platelets within the lung. This may lead to produce various effects, including bronchoconstriction, involvement in the late asthmatic response. Leukotrienes C4, D4, and E4 liberated during inflammatory processes in the lung. This leukotrienes produces bronchospasm, mucus secretion, microvascular permeability, and airway edema. Platelet activating factor is involved in the mediation of bronchospasm, edema formation in the lungs. Next slide, 10. Role of adhesion molecules in airways inflammation. An important step in the inflammatory process is the adhesion of the various cells to each other and the tissue matrix to facilitate the infiltration and migration of these cells to the site of inflammation. Adhesion molecules have additional functions involved in the inflammatory process aside from promoting cell adhesion, including activation of cells and cell-cell communication, and promoting cellular migration and infiltration. The many adhesion molecules are divided into families on the basis of their chemical structure. These families are the integrins, cadherins, immunoglobulin supergene family, selectins, vascular addressins, and carbohydrate ligands. Adhesion molecules are found on a variety of cells, such as neutrophils, monocytes, lymphocytes, basophils, eosinophils, granulocytes, platelets, endothelial cells, and epithelial cells, and can be expressed or activated by the many inflammatory mediators present in asthma. Thank you for your attention. Further information, kindly contact profsamiacademy@gmail.com. Disclaimer: this channel does not promote or encourage any illegal activities, and all contents provided by this channel are meant for educational purposes only. Any copyrighted material on these slides is included as fair use for educational purposes only. It will be removed at the request of copyright owners.